Okay, I think we'll go ahead and get started. So this is the Release 66 webinar, and as if you've joined these before, hopefully they're familiar to you. Our goal really is after each of our releases, which we have once every 2 months, our goal is to give you a quick rundown of what's new in our resources, and which is kind of nice. It gives you an idea both on what new data is available, what new functionality is available, and we demo a bunch of the things. So it ends up being, both a combination of informative in terms of what's new, but also informative in terms of what you can actually do on ViewPathDB resources. My name is Omar Harb, and I'm based at the University of Pennsylvania, and I'm going to be joined today by my colleagues Gloria Giraldo Calderon, who's based at the University of Notre Dame, and Evelina Besenko, who's based at the University of Liverpool. And we'll sort of tag team in terms of presenting different aspects of the resources. Eve will focus on covering what's new in FungiDB, and Gloria will focus on covering what's new in Vectorbase, and I will cover some cool or new features and data sets in the other resources. Hopefully you're familiar with GoToWebinar or many of these online tools, but there is a question and answer panel that we encourage you to use to ask questions throughout the webinar. We will monitor those questions and answer them either live or just by typing back the answer. Also, this webinar is being recorded, just like all of our webinars. And we do have a link here to the webinar page, but again, don't worry about trying to remember this link. I will show you right now. I'm just switching to our browser. And so if you take a look, and I go back to the ViewPathDB homepage, under Help, there's a section called Learn How to Use ViewPathDB. And under Learn How to Use ViewPathDB, you'll see a number of things. These include links to our frequently asked questions, but also our webinar page, which you can explore. And you will notice here there are different kinds of webinars. We have these on-demand training webinars, and those are specifically covering training topics, like how do I use a strategy system, how do I do Go enrichment, and so forth. But also we have release webinars, which are collapsed by default. And you can go in here and you can see all of our release webinars are linked. And the Release 66 webinar, by tomorrow or the day after, it'll show up here as well. So you can always come back and review these webinars at a later date if you want, or if you missed something. Just pointing out that the help section also includes a section for workshops. Some of you may know that we had a virtual workshop last week. If you missed that workshop or weren't able to attend the whole thing, you can go to the workshop page, and you'll see under previous workshops, there's this virtual workshop link. And when you go here, you can go down to the schedule, and you can basically take this course asynchronously. We have recordings, of all the joint sessions that were worth, recording. They're all there, and associated exercises that went with those sections are available here. These will walk you through the various, step-by-step walkthrough through the exercise, and you don't necessarily have to be part of a workshop to take these. And so I encourage you to check these out and maybe watch a YouTube or the link to the presentation, and then do the exercise as well. As usual, if you have any questions, whether it is about any of the workshops or the webinars or the functionality of the site, just click on the Contact Us link. This is a really easy way to get in touch with us. Fill up this form, write your question. The outreach team gets your emails, and we try and respond fairly quickly, depending on the load, workload that week. But typically, we try and respond within 48 hours. I wanted to point out before we go into what's new in our resources is that we do have some upcoming workshops that you may be interested in. We have, and both of those are in-person workshops. We have one that is a general ViewPathDB workshop. That's our annual workshop. I think this is maybe our 15th annual one or 14th that we've been holding in Athens, Georgia. And this is going to be taking place in May, and you can learn more about the workshop and apply. We also have a fungal pathogen genomics workshop coming up in June. This takes place in Hingston in the UK. And again, the this deadline is coming up soon too, and I would encourage you, if you work on fungi, this may be an interesting workshop for you to apply to. These get fairly competitive, so apply soon, and obviously make sure you're answering all the questions fully, because they do take those answers to those questions carefully into account. Okay, so what's new in release 66? So release 66 came out a couple of weeks ago. And if you ever are wondering, what is new in release 66 or any of our releases, you can go to the news section, which is right here on the right-hand section of the website, of any of our websites. And you can click on the section here, and you'll see that there's a news section, and you can click on see all, or you can select a specific one depending on what you're interested in. I like to go to see all because it lets me quickly scroll down and go through these. So each of our resources has its own news section. And so you can go in, if you're on the ViewPathDB main page, right, you will see the news section for all of them. But if I go to, let's jump to TrickDB, for example. And so this is the database that focuses on Trichomonas vaginalis mainly. You'll notice it also has, and hopefully you're aware of this, but all of our sites are built in the same infrastructure. They look the same, they function the same. The only difference really between the sites is going to be the type of data, and obviously the genomes that are available. So Trichomonas will include trichomonatids, and so that'll be the big difference. But if you go to news section here, you'll see that there's only TrickDB news. So again, depending on what you're interested in, I would, you can access these in different ways. Okay, so while I'm on TrickDB, I'll start with TrickDB. This is, I think, a very nice release for Trichomonas researchers, because as with many of these organisms, depending on the accessibility of the organism and what technologies are available, so some of these organisms lag behind in terms of availability of data. But this release was very nice for Trichomonas, because there are a couple of new RNA-seq data sets available. There's a ChIP-seq data set, so looking at histone modification, various histone marks, it's a brand new data type for Trichomonas. And also there's a proteomics data set for extracellular vesicles. So really covering 3 different data types in this database, so that's kind of nice. So each of these data sets, and you'll see this when Gloria and Eve speak as well, each of these data sets has its own data set page. So I can click, for example, I'm interested in this Trichomonas vaginalis response to metronidazole. I can click on this. This will take me to the data set page. So this is a very specific data set page for this particular data set. You'll see there's a link to the primary publication. There's information about who the primary contact is, when we got this data, and so forth. As you scroll down, again, we have a section with a link with the publication, all the sort of collaborators and contributors to this project. A short description. Sometimes this is very extensive, sometimes it's minimal. We rely heavily on on the people who generate this data to give us this information. And so, if you notice that a description is lacking and you've read the paper, feel free to drop us an email and say, hey, you know what, for this data set, I think if you can add this information to a description, it would be really helpful. And we'll be happy to do that. This is super easy for us to do. And of course, getting input from the community is great. When you scroll down on this page, you will notice a couple of things. We have a section for exploring this data, and this will link you to wherever this data is present, whether it is in a genome browser view or searches available against the data. We have access to those here. And then also we have an example. In this case, it's an RNA sequence data set, so we're looking at an example graph. So this is a gene, I don't know what this gene is, but this gene has, it looks like, maybe a 3-fold upregulation in metronidazole treated samples compared to the untreated samples. But this is just an example. I think that these are just random examples. So sometimes there's no differential expression depending on what sample was selected. All RNA sequence datasets can be accessed in the same way on all of our sites, and I'm sure you'll see this repeated when even Gloria highlights some of the datasets in FungiDB and VectorBase. But you will notice that for this particular dataset, there are 3 types of searches. There's a fold chain search, differential expression, and percentile search. most of our data sets will get, if they have more than one condition, they will get a fold change search and a percentile search. And differential expression typically is available for data sets where there are biological replicates, so we can use statistics. Nowadays, most of the data sets will have biological replicates, and so you will notice more and more of the newer data sets have all three of these types of searches. And there are additional searches that are available, but we won't discuss them today. There are other webinars that cover those in more detail. But if I'm interested in finding all the genes that are upregulated during metronidazole treatment, I can go and run this differential expression search. So if I click on this link right here, it takes me to the specialized search page for this particular data set. And I can select, I want to compare metronidazole to the normal cells. So we always, our comparators, always the numerator and the reference is the denominator. So if you like to think about equations, so this would be on top and this would be on the bottom. And I can ask for genes that are upregulated, and I can change this to, let's say, 4-fold upregulated. I can play around with the p-value and make this a 0.01, for example, or we can play around with this, and I can run this search. And so this tells me it found close to 3,000 genes that met these criteria, right? That's a high number of genes. I may decide that, well, you know what, maybe I should be looking for things that are even have a higher fold change, and that's fine. I can revise this step and can go, all the parameters will still be there that I selected, but now I can go in and change this, let's say, to 8-fold, for example, and maybe change the p-value to 0.001 and revise. And, so this number went down as to be expected. You can go and explore these genes and look at these fold changes. Okay, so for all of our releases, we also introduce new features. Not every release, but in many of our releases, we have new features. And so one new feature that we introduced in this release is a additional download options. And so all of our data is downloadable. Anything you access in the sites is downloadable in many different forms. For example, I can download this entire list of results, this 1,355 genes, by clicking on this download link, and then you'll have different options that you can explore. Or I can go to an individual gene. So if I go to this individual gene page, so this is going to load this encyclopedic view of this particular gene. I can take a look at it in many different ways. I can look at the transcriptomic section, for example, and look at the RNA-seq expression section. There are a couple of sections that are of interest here, and I can find the one that, for example, looked at the metronidazole expression, and I can explore it further and go from there. You'll notice this is an interesting gene here. You'll notice there's this unique and non-unique, right? So now this is actually gives me a little bit of a pause here, because this tells me that there's a lot of non-unique aligned reads for this particular gene. So it's hard for me to tell whether this gene is truly differentially expressed or it's one of its paralogs. And you'll notice that we'll have a warning here. This gene has 19 paralogs, right? So this gene has 19 duplicates probably, and this is a common thing in Trichomonas. There are many gene duplications, which makes it a little bit harder sometimes to interpret some of this data. So it's just something you have to keep in mind as you analyze this data. Okay, so the feature I wanted to highlight to you is some new download feature. So if I go to the top of the gene page, I can click on download gene, and now you'll notice that you have you can download text, which is basically all the tables on the gene page, and I can select whatever I want, and you get those tables. But now we have this bed format. So if you want to download something that you can load on another browser or view it in different ways, so you can download different aspects in bed format. And you can also download this FASTA. You can always download a FASTA. This was available in the past as well, but we have additional features, especially for genomes that have introns. This may be quite interesting because you can actually include or exclude introns in your search, and in any case, many other types of sort of formats of your data that you can download. And all of these, you'll notice that as I click on these, you get different options given to you. So for example, if I want to download the protein features associated with this protein, right? Let's say all the intraprodomains, I can do that. I can select that, and if this had an intraprodomain, I would be able to download it. Okay, so I'm gonna quickly switch back to ViewPathDB, and let's go back to the news section. So scrolling down, again, I'm not going to go into every single data set. There's a lot of data sets, and it would take us a few hours to cover them all. But to highlight, if you work on trypanosomatids in TriTripDB, we loaded some really cool data from Carlos Rebello's group in Uruguay, and those include transcriptomics, RNA-seq data sets for different stages of Trypanosoma cruzi. We've mapped these data to different organisms, but also it included ChIP-seq data, so chromatin immunoprecipitation data, using various methylation marks. And so that's kind of interesting because you can see then accessibility to transcription across the genome. And again, with all of these, you may be curious, well, I really don't know anything about these methylation marks, or methylated sites in the DNA, how can I learn more about it? And that's where, of course, going to the paper directly, and that's something I would highly encourage. In fact, it's something I was doing earlier. I had to go in and say, oh, I can't remember what these do. But you can go in and look at the paper and learn from the paper exactly what they did, and then come back to ViewPathDB and explore the data for your genes of interest. I'll take a pause here to point out that we get the question frequently as to how do I cite, when I use a data set in the database. And so we always encourage you to do two things. So one is, obviously, if you use the database and it was useful for you for your research, definitely make sure you cite the database. And if you're curious on what publication to use, you can go into our sort of about section, and you will see that we have there are different. First of all, there are different sections here that you can learn about what how data is being used or how these resources are being used. But also, if you go to what is ViewPathDB, you'll see a section there with a link to the most recent publication. If you need to go on our website to find it, but you can obviously find those publications on PubMed as well. Of course, we also encourage you, and in fact, you probably should and must cite the original authors who generated this data. So always do that. So I would encourage you to do both. So cite the authors that generated this data, and that you access this data then through ViewPathDB, and you can cite the ViewPathDB resource. Okay, so moving down, I want to move on to ToxoDB. So ToxoDB, besides having, again, some really cool data on Besnoichia besnoichii, which is another apicomplexa that infects cows. But again, just like Toxoplasma, it has its tachyzoids and bradyzoid stages and so forth. And so this is very nice for comparative reasons. So you may be interested in knowing, well, out of these genes, maybe I'll highlight this very quickly. I'm just going to go to this to the dataset description page, and I'm going to run a full change search for this dataset. Give it a second to load. Actually, I should probably be doing this in ToxoDB. It's a little bit faster if you do it on ToxoDB. So we'll just go to ToxoDB, and I'm going to show you this is a different way of getting to the data. So I'm just going to go quickly to the RNA-seq data. These are all the specialized searches. The same searches are available here on the left, but also in the middle here. I'm going to click on RNA-seq evidence, and now going here, and you'll notice any new data set for each release gets a new tag on it. So I quickly can see that, oh, these are two new data sets, so I don't really have to search. But if I really wanted to search anything that is only in Besnoichia, I can just start typing Besnoichia, and you can see that. Or I can start typing the name of the author, for example, and then that will filter down to that. But here's this particular data set. I'll go to the fold change query. And right here, I'm going to compare 8-hour infection to the 4-hour infection and find anything that's upregulated by 4-fold, for example. And so this gives me, it'll give me a certain number of genes. So there's 36 genes that met this criteria. I can look at them and explore them. But what's really cool is you can ask, well, do these genes have homologs or orthologs in Toxoplasma? Because you're interested in knowing if those homologs have a similar expression pattern in Toxoplasma. So I can add a step and use this transformation step right here, transform based on orthologs, and now I can select Toxoplasma gondii, and I'm going to pick the MA49, which is the reference strain, and I'll run the step. So what this is doing, basically, it's saying, well, for any gene that has any of these 36 genes that has a homolog, in Toxoplasma bring back that homolog in Toxoplasma. And so it tells me that there are 53 homologs of this gene, and here they are. And so you may be interested in looking at the expression pattern of these genes and seeing if there's something similar between them. And that might actually be able to, it allows you to leverage data across species to identify potentially some interesting things in the species of interest. While I'm here, you'll notice I have a search strategy. This is something I was running before. Maybe it was part of the workshop last week. And you'll notice there's this one step that has a big X in it. So this means that this step is no longer valid. And that's because something changed with this query. Something changed in the parameter, which means you have to rerun this search in order for this particular strategy to be valid again. And often all you have to do is click on edit. And when you do that, it gives you, we preserve the information. And so you can go in and say, oh, that's what I selected. So I just need to go in and make sure I select the same samples and run this again. And there are many different reasons why this would become invalid. This is an example where I think it should have run automatically and fixed the issue without you having to rerun it. So I'll follow up on that. But often, we may change one of the parameters, or we improve the way the data is being represented or analyzed, and that ends up basically tell you that, look, you need to rerun the step, make sure that you still want to use the same parameters as you did before, instead of just giving you results that may not be meaningful anymore. Okay, another big thing in ToxoDB for this release is that the main reference annotation was updated, and so I'm just going to go back to the news section. This was a huge community effort that went on for years, actually, and included both integration of community knowledge using user comments and also using the Apollo system, which Gloria will tell us a little bit about in a few minutes. But as a result, and working with the community, the annotation of the ME4RN genome has been updated dramatically. And if you're interested in knowing, well, what are those changes, you can go to this spreadsheet right here. This will open up, it's just a Google spreadsheet with a number of tabs here. And so you can quickly go in and say, oh, wow, they're new genes. And you can look at this, and they're like close to, I think, over 200 new genes that were annotated. And so that's kind of exciting, right? This is a genome that's been around for a while, and it's still, we have new genes, or genes that got split, or genes that are merged, right? And so there are many different changes that happened, and this will be, obviously, if you work on Toxoplasma, it will be super important for you to explore this in detail. This improved annotation, I think, is really, really much better than before, even though it wasn't horrible before, but this is a big improvement and an exciting one to be able to provide to you. And again, if you have any questions about this, just drop us a line and we'll be happy to answer them. So, okay, so the other, let me go back here, and let's take a look at read evidence. So one new, so we started integrating long-read RNA sequencing data. So presumably every transcript or every read that we get is a full transcript, and so this allows you then to start looking at isoforms of transcripts. And we integrated a new search called the long-read evidence search, which is available here. So depending on the data type, the data we have, you can go in and run this query. I'm not going to go through it in detail. There are lots of parameters here. They're all described here. This is still in beta, which means it is a search that is going to most likely change over the next couple of releases as we get used to it and learn exactly how is it functioning appropriately. Do we have any issues with it? But also learning from you, whether this type of search makes sense. Do we need to add additional help? Do we need to change the descriptions here? But the bottom line is you can go in here and you can start asking for genes that have novel isoforms that don't exist in your in the annotation, and this may then hopefully enable you to identify isoforms of genes of interest that may end up having some interesting function that you want to explore further. And you can go and explore these in the genome browser and so forth. Okay, so I'm gonna jump back, and I'm sure there's a lot we can talk about the improved annotation and of course the long read RNA sequence data. So very quickly, I want to make sure I leave enough time for Eve and Gloria. So let's move down quickly. I told you about the updates in ToxoDB. I've told you about the two new features, the long-read RNA-seq feature and also the new download feature. Point out in PlasmoDB, we added a new Vibex genome. We also added a number of transcriptomic data sets for both Falciparum and ULAI. So again, if you work on Plasmodium, it might be interesting for you to look at those. In particular, these data sets are gametocyte data sets, so that will be kind of interesting. And these are nanopore long-read RNA-seq data sets. So probably something very cool to explore and see if there are any isoforms that are unique to gametocytes that are not present in other stages of Plasmodium. Notice that we have updates to the functional annotation. This is a continuous effort. And for every release, almost every release, you will see that their genes, that their functional annotation, meaning the description of the gene, what the gene does, has been updated. And again, there will be a summary table that you can explore further and see what all of these updates are. And you can, obviously go into more detail and look at them. Sorry, this page is just still loading, so we'll leave it here. Let me move down. PyroPlasmaDB, again, so this is a database that includes Babesia and Theileria, but there's a very cool data set looking at a time course of expression of Babesia diversions. And so if we look at that data set again on PyroPlasmaDB, I won't run it. It runs the same exact way as all of our other RNA sequence data sets, but you'll see here, there's this data set, and if I go to the fold change query, I can quickly go in, and this is, I was playing around with it earlier, but I can select different time points, compare them to each other, set my parameters and run my search, and you'll get a list of genes that meet the criteria. Okay, moving down, HostDB, there are a couple of data sets here. I won't talk about them. They're both transcriptomic data sets, so you run them exactly the same way. Just to say that these are basically data sets of host response to pathogen infection. We have one that is a host response to Bisnoichia infection. So remember, Bisnoichia is this cow pathogen. So this is going to be cow genes that are upregulated or downregulated in response to infection. But using that same orthology tool that I showed you in Toxoplasma, you can actually then say, well, are there homologs of these genes that are differentially regulated during a Bisnoichia infection in a cow? Are there homologs of those genes in humans, for example? And then you can explore those and see if there may be some interesting things in there for response to apicoplexa in a human infection. And then there's also another infection or host response experiment from Aspergillus fumigatus, and maybe Gloria, maybe Eve will talk about this some more. But suffice it to say, you'll access this data the same way as you access the transcriptomic data in Toxoplasma or in Plasmodium and PlasmoDB. Giardia just got a new update to one of the genomes. I'll scroll through FungiDB, and just to point out that CryptoDB got some updates. In particular, we've been doing this automated way of improving the annotation by adding PFAN domains. And so if a gene is annotated as a gene with unspecified product or unknown function, we have a way now to automatically assign it with, and if that gene has a domain, a functional domain, so let's say a gene has a kinesin domain, then this gene will become a gene that's hypothetical but has a kinesin domain. Its new name will be something like kinesin domain containing gene or something. So it's a little bit more informative and allows you to see right at the top of the gene page some useful information. And finally, same thing with AmoebaDB. It got some updates to the annotation of the PFAN domain. So a couple of Naegleria genomes got these updates. So I know this was a whirlwind of going through all of our databases. We have 2 more big databases to go through. So I'm going to stop here, stop my sharing, and Eve, I will switch it to you so you can cover FungiDB.
Okay, thank you, Amar. Let me just share my screen really quickly.
I'll make you the presenter. Oops, sorry. I muted you by mistake. Hold on.
It's all right. I got it. Yeah.
You got it? Okay, good.
see. Well, you should be able to see my window, which is on the FunGTB news page.
Yes.
Great, great, thank you. Okay, so in the next few minutes, I'll walk you through some of the updates that we have had on FunGTB in this release. As Amar have mentioned, FunGTB is running a workshop called Fungal Pathogen Genomics Workshop next year in Hinxton, United Kingdom. So if you have never heard of this workshop before, this is a collaborative workshop that involves several databases, including Ensemble Fungi, Saccharomyces Genome Database, Candida Genome Database, and also Microcosm JGI, in addition to FunGTB. Here I'll show you really quickly one of the page where the workshop is advertised through the Welcome Connecting Science that organizing this workshop, that provides a little bit more information about what the workshop is about. So if you're interested in learning and how to use these web-based data mining resources to answer your biologically relevant questions, whether you're working on fungal species and oomycetes, this may be a workshop that you are interested in. The program includes a number of various interesting topics that deal with navigation of the sites and also using web-based resources and various bioinformatic tools to mine data integrated in the databases. So for example, here's an example of the workshop that we have ran earlier this year in May. And the workshop included several sections, such as pre-workshop modules that work you through the navigation of the databases, and the in-person workshop module includes hands-on training exercises on how to mine data in various databases that I have mentioned. And that comes also with an additional modules on how to analyze your own data in Galaxy. And we specifically looked at the transcriptomics, such as RNA-seq data, and also cold SNPs on the whole genome sequencing of isolates. So coming back to FunGTB, so I want to also walk you through a few new datasets that we have had. For example, there are several new datasets that that we have loaded, and we have also replaced 3 genomes. This has something to do with integrating new genomes and also syncing with NCBI records to make sure that we have the up-to-date records. So, for example, Coccidioides immitis was reloaded to integrate the latest annotations available in NCBI. For when we can, if there are any warnings or issues detected by NCBI, we also try to let you know. For example, this particular genome is now scheduled for reloading because based on the authors and also note from NCBI, there are some issues with contamination of this genome with other reads from another organism. So we have scheduled this genome for reintegration in the subsequent release in 2024. And as Amar had mentioned, we have also updated in FunGTB gene products, where unspecified product was replaced by more sort of intuitive description of the product using PFAM domains. And this has happened for a number of organisms that are listed here below the annotation update section. In terms of the functional datasets that we have integrated, we have several datasets to share that includes transcriptomics and also SNP analysis. For example, for the transcriptomics data, we have 1 dual host pathogen datasets for Aspergillus fumigatus. And this particular dataset has experiments where Aspergillus fumigatus strains were incubated with human epithelial cells A549. So in this case, this particular data set has 2 strains of Aspergillus fumigatus, and Aspergillus fumigatus causes aspergillosis, which is a significant human disease. And in this case, 2 strains were incubated with human cells for at 2 time points, and the RNA-seq was performed to investigate how gene expression changes at these criteria. So, for example, if you wanted to access any of these data sets, you can click on the dataset link, which is, as Amar have shown you, is highlighted here in blue, and I have already opened it separately on my screen. And so you will be redirected to a dataset record page that provides additional information about the experiment, also the authors that have published the paper, and provides a number of the searches that are available for this dataset. In this case, I have identified this dataset in FIND-GTB that hosts the fungal component of the data, and I have also navigated to HostDB, which holds the host component of the data, because the cells are belong to the human airway epithelial cells line, the reads have been aligned to the reference genome for humans. And so in this case, having these 2 data sets available for essentially data mining, you could envision setting up a search that may look for genes that are regulated in Aspergillus fumigatus and also in the host, such as human cells, in response to the infection. And so as I'm scrolling down the page, notice that there is a representative example graph for the different samples, and also a list of the searches that you can employ for this data set. So if I scroll a little bit, see, for some reason they're closed off, but explore this data set, right? So if you want to explore this data set further, you can expand the explore this data set within the website to bring up additional options. So if you want to look for upregulated genes, you can click on the fold change, and you could bring up the interface that will allow you to set up your experimental conditions. For example, I'm interested in looking for upregulated genes, and I'm using a 6-hour sample as a control, where it was just the epithelial cells that were growing in media for 6 hours. And my comparison sample will be those cells that were exposed to the AF293 strain of Aspergillus fumigatus. Notice that the section on the right gives you a graphical representation of your search criteria in terms of the expression values as they correspond to the reference and also the comparison sample. And so if you click on the get answer, you will get results for this type of search. I set up this type of search both in FIND-GTB and also HostDB. And here I'll show you the results for this search. So here's the results for FIND-GTB, and here's the results in HostDB. So the criteria that I have used were essentially the same. I have looked at, in FIND-GTB, I have looked at the cells that were incubated with human epithelial cells and determined those genes that were upregulated by at least 2-fold. And in HostDB, as I have just shown you, I have looked for genes that were upregulated also by 2-fold, but in the host when incubated with Aspergillus fumigatus. And notice that I have identified 11 genes that fit the criteria within the host, and also 288 genes that were upregulated in Aspergillus fumigatus. And so from this point on, you can explore genes further. For example, if in Aspergillus fumigatus, you can either combine this search with additional criteria, such as bringing another transcriptomics data sets or SNPs, or even your own data if you have a list of genes. And you can also further analyze your data by enriching genes either for GO terms or metabolic pathways to see perhaps there are some interesting patterns that can be observed within this data set. And if you're in HostDB, you can similarly mine this data set by looking at the genes that are upregulated within your conditions. And if you want to look at the graphical representation of the data, it will be represented to the right within the results table. Notice that here I scroll down to the right, and now I see the expression pattern of each of the genes that return, also showing not only for the specific sample that we have chosen, but also across all of the samples that exist for this data set. So, and the same will be true for the Aspergillus data if you return to the gene results page and essentially scroll to the right within the results table. Right. So at this point, I'll go back to FIND-GTB News and show you just a few more additional data sets that we have integrated. So one thing that I want to mention is the Aspergillus niger co-expression network. And in this case, the authors have analyzed over 155 transcriptomics experiments where they have looked at how genes behave in terms of their expression analysis, and they have identified genes, those that are co-expressed together, and now these data can be accessed and mined within FIND-GTB. So to identify genes that are co-expressed, you can simply navigate to the co-expression search. And in this case, I'll be using the default selection where I'll look for a specific gene ID within Aspergillus niger. And what I will be looking for the genes that have a positive correlation and with Spearman coefficient at least 0.5 or greater. So I'll click on Get Answer and wait for the results to return essentially with any of the genes that match my criteria. So in this case, I returned 20 genes that are associated with this particular gene. So they're co-expressed in a similar way. Notice that there's a number of genes also with protein of a known function, while others have known orthologs, but yet do not have a product description listed. So this is, in this case, you may want to look even further. You can combine the observations for the strategy with additional searches such as AlphaFold prediction, and see if for any of the proteins of an annotated function, you can actually determine any folds that are picked up by AlphaFold, and as such, you can learn a little bit more about their function that way. AlphaFold search is now available in FIND-GTB as well. So if we look for, click on the Add step and then look for AlphaFold amongst the available searches, you can deploy it from the drop-down menu. And since we're working with Niger, I'll select Niger from the reference genomes that I want to run the search on. And by clicking the Run step, the 20 genes that are in my search will be overlapped with the records provided by the AlphaFold. And here now I have that out of the 20 genes identified in step 1, 19 of them have known AlphaFold predictions. And so in this particular case, you may want to
I don't think we can hear you anymore. Can
Yeah, no, I cannot hear and just see a black screen.
Okay.
Should we wait for a second, or do you want me to go?
I think you can go ahead, Gloria, since we only have 15 more minutes, and I think it's probably a good time to transition, and then Eve can say anything if anything comes up.
Okay, good.
Made you the presenter.
Thank you. Okay, are you able to see my screen now?
Yes, we can.
Okay, excellent. Thank you. Well, as Eve and Omar already show you, this is all the sites work exactly the same. So if you come to Vectorbase and click here on this panel, select the release 66. These are the highlights for Vectorbase. In case of genomes, we have 6 new genomes, and one of them that I want to highlight is this one, Sabetes cyaneus, which has been called the most beautiful mosquito. And if you have never seen it in person, I encourage you to Google an image. It's iridescent, like a hummingbird, and has feather-like legs, and has a really nice, the male does a courtship, again, like a bird. So it's a beautiful, beautiful mosquito. And now we have the genome. The people that sequence the genome have open for anybody who wants to analyze it. They just are going to publish a note in which they're basically just saying, here is the genome, but not dwelling into any of the specific gene families or anything. So this is completely open for the community to use the genome and analyze it. And if you are ever in doubt with any of the genomes that we release, that which one can you actually use for anything, or if the the data providers are actually working on a genome paper, please always click here, contact us, and let us know. Okay, other genomes that are also open for you to analyze are Anopheles marshalli and Neely to generate any kind of papers that you might want about them. The snail, Biomphalia glabrata, we already had a genome for this species, but we got this new strain which with a much better assembly. And we also have two sandflies, Lutzomia longipalpis and Phlebotomus papatassi, also a great upgrade from the previous genomes, and the data providers are working on the genome paper right now. So hold your analysis for now, or just, you know, working on a few genes, but genome-wide analysis, hold on for these sandflies until the data providers, you know, get their own paper. We also have a genome update. The scientists from this group generated their own annotation and they uploaded to GenBank, and that's what we reflect right now. This one that Omar mentioned a little bit in one of the other parasite databases, I think was Toxoplasma that Omar said. So yeah, we are very excited when users come to our databases and they decide to annotate genes, because you are the experts and we are very happy to help you put those annotations in Vectorbase. In this case, there was a team of scientists, and here we have provided their names and their institutions, that came to the genome of Culex kinquefasciatus, Johannesburg 2020, and annotated 425 transcripts corresponding to 391 genes. And here in this graph, you can see how many of those genes that they look at, they decided that needed modification. They added completely new genes. They deleted some that there was no evidence, RNA-seq evidence, or really they were not good predictions. And some genes, they just decided, you know, the automatic annotation already provided looks good, split some genes that should be 2 or more, and merged some gene models that should become 1. And these genes were basically odoron binding proteins, odoron receptors, and here we have this cloud with the genes that they annotated. If you click here, you will see the list of genes that they annotated. Is the 391 genes. Here we have the gene name or symbol, the product, and as Omar already explained, you can download the list and know what is new for this genome. We not always acknowledge with this such detail who annotated genes when you only do 1 or 4, but we always are very happy for every release whenever we get you know, new submissions. And this gives me the point to talk about Apollo. So if you come to Tools, Apollo, and this is the tool that people use to share those annotations with us. And this page is now different if you are familiar with it. Here you can get an image of what to expect from Apollo. How does it look with the evidence, RNA-seq evidence, the orthology, and the gene models that you can modify. If you are already an experienced user, click here. But if you've never seen this before, this is a practice, play, sandbox area. Nothing that you do here will remain will affect the gene set, only the ones that are here. Here we not even look at the sandbox. This you know, you can do anything you want with any genome. And here we have what is Apollo and different types of documentations that you might want to take a look if you want to annotate some genes. Either if it's just a few or just a team effort, we're happy to hear from you. And we also got RNA-seq evidence. We got 6 new RNA-seq data sets, 4 for ticks and 2 for sandflies, again, Lutzomia longipalpis and Phlebotomus papatassi. And already, as Omar and Eve demonstrated, we have the data set page where you got the reference, the people who generated data, the type of searches available, and you can see for any random gene, you know, how is the expression of that gene. So I'm not going to go over that again because we already spoke about that. We also have the map which you can access from here, Tools, Map View. We also have a beta version because we are working on improving this. And the map has more than 700 studies, and these are the new ones for this release. If you are not familiar with our new map interface, I encourage you to come to our workshop from last week and look at the presentation. You can come to help, learn how to use ViewPath, workshops, and as Omar already demonstrated, come here. And the very last one of the talks was about the map. Here, map view introduction. So I encourage you to watch this release. And I think that's about all I want to cover. And here's what I just said. We are always happy to hear from you and learn, which genes you might be interested in annotating. And maybe if it's 1 or 2 genes and you are not up for, learning how to use Apollo, just contact us and tell, okay, this is my gene of interest. This should be the name, this should be the product description, and we will do it for you. And that is all I want to say.
Great, thank you, Gloria.